Very good morning to one and all. I, Dr. D. Lakshman Kumar, Associate Professor, Chebrul Hanumaya Institute of Pharmaceutical Sciences, Gunduru. I take this opportunity in starting the first session of day 3 on AICTE and ISTE sponsored induction program on training on pilot plant scale-up techniques, phase 3. On this note, I would like to introduce today's speaker, KV Ramanamurthy Garu. Sir is retired professor and principal, Andhra University College of Pharmaceutical Sciences, Andhra University, Visakhapatnam. Sir's education, Sir completed BPharm in 1979, MPharm in 1982, PG Diploma in Applied Statistics in 1983, and PhD in 1987 from Andhra University, Visakhapatnam, Andhra Pradesh, India. Academic experience, he is having more than 35 years of teaching and administrative experience. Awards, Srishti Gandhian Young Technological Innovation Award as mentor in 2019. Awarded State Best Teacher Award in Pharmacy by Government of Andhra Pradesh in 2015. Awarded Dr. Sarvepalli Radhakrishnan Best Academician of the Year 2010 by Andhra University. Awarded APTI's Teacher of the Year Award 2009 by Association of Pharmaceutical Teachers of India. Awarded AP Scientist Award 2007 by Andhra Pradesh State Council of Science and Technology. And he had several travel grants from various government organizations. Professor Ramanamurthy sir, areas of research interest, controlled release dosage forms, development of oral lipid-based formulations, development and evaluation of vesicular drug delivery systems, chronotherapeutic drug delivery systems. Sir had guided several research scholars. Sir is having one postdoctoral student from Nigeria, and Sir awarded 64 PhDs and Sir guided 141 MPharm projects. Sir have published 172 research papers in various national and international journals, 95 research papers presented at various scientific conferences, 12 research projects completed. Sir has 3 patents. For his credit, he served as Principal, AU College of Pharmaceutical Sciences, Chairman, Board of Studies, Andhra University College of Pharmaceutical Sciences, Visakhapatnam, Member, Consent for Operation, AP Pollution Control Board, Member, NAC Steering Committee, Andhra University, Placement and Training Officer, University College of Pharmaceutical Sciences, Andhra University, Visakhapatnam, Membership in professional bodies, life member of professional bodies like Indian Pharmaceutical Association, Association of Pharmaceutical Teachers of India, Indian Pharmacy Graduates Association. With this brief note, I request Dr. KV Ramanamurthy Garu to kindly take the platform and to start the session. Over to you, sir. Sir, please unmute and start, sir.
I hope you are able to listen to me, right?
Yes, sir.
Yeah, thank you for your nice words spoken about me. And I think I'm trying to share my screen first. Yeah, I hope it is visible to everybody.
Yes, sir.
Yeah. As part of the training program sponsored by All India Council for Technical Education, today I would like to share some of the views related to pilot scale-up. So whenever a research project that is developed in a laboratory may be called as a research curiosity by the researcher or a successful manufacture on a high-speed production equipment with a cost effectiveness. So one should be able to differentiate whether it is an academic research or an industrial research in the normal opinion. So majority of the institutions in India are developing the academic research products. That means they are getting very good results in achieving their targets. But majority of these products that are developed in the laboratories are failing to reach to the industrial production due to various constraints. Whereas The foreign universities, especially the US and UK and German universities, are very much in collaborating with the industries, and they discuss what the industry is in need of it and how that is to be manufactured right from its developmental stage to its production stage. So before the product reaches to the industrial scale, it must be thoroughly tested for its robustness for withstanding the batch-to-batch variations, process variations, and modifications, and it should have sufficient stability and process parameters that are developed for that. So the problem is how we can transfer these results of the laboratory scale, which is at the R&D level, to the production scale, which is always maybe linear. Sometimes it may not be linear for the large scale production, altering the final product specifications. The meaning of my linear is, for example, if you are having a 100 tablet batch, you just simply increase it to 1,000, 10,000, something like that. Similarly, we can increase the speed, or we can increase the volume of the same equipment. But many a times, this linearity may not hold good properly. So to overcome this, an intermediate pilot scale batch is to be developed, which is going to assist to study the problem of the scaling up to the industrial production. So what is the definition of a pilot plant? It is defined as a part of the pharmaceutical industry where a lab scale formula is transformed into a viable product by the development of liable practical procedure for manufacture. So it involves manufacturing of drug product by a procedure fully representative and simulating that is used for full manufacturing scale. So scale up is defined as the act of designing a prototype using the data obtained from the pilot plant model. So there are three different stages before we reach to the final production. The first one is being R&D, second one is the pilot, and the last one is the production. So what is the significance of this pilot plant? It is to identify the problems of small scale and solve these problems during large scale production for making profit. So the objective is to produce physically and chemically stable therapeutic dosage form. We need to review the processing equipment that is going to be used in the production, and we need to follow the guidelines for production and process control, which is very important nowadays. Whenever a document is being filed for approval, the first question that is being asked by the regulator is, what guidelines that are followed by you? That means the guidelines that are stipulated by the respective regulatory agency must be specifically followed during this entire operation of pilot scale-up and production manufacturing. And we need to create the evaluation and validation documents, and we need to identify the critical manufacturing variables of the process. And at the end, the pilot scale must be capable of producing the master formula record, which is going to be used throughout the production of the product. So what are the different stages of pilot plant? So we need to examine the suitability of the laboratory formula for the production. What are the different processing equipment that are necessary for the production? We need to define the specifications of the raw materials and the production rates, very important. Without specifying the specifications of the raw materials, it is practically impossible to scale up a process, because the raw materials may have different physicochemical properties. For example, the particle size, which plays a very important role during the industrial scale up, is very important. And if the raw material supply is not sticking to the specific particle size as required, the production batches may alter their assay and other parameters. Assessment of the requirement of the physical space. So what is the requirement of the space for this production? And appropriate records and reports which are going to support the GMP requirements. So we have a good proverb, one size cannot fit all. So lab scale preparation equipment often cannot be applied for large scale production. For example, in the laboratory scale, for production of a nanoparticles or a microparticles, magnetic stirrers are the most commonly used equipment for stirring, but that cannot be magnified to the industrial scale of production. Similarly, the scale-up conditions may also change with respect to the application of the situation that is required in the scale-up. For example, mixing applications are concerned with increasing the linear dimensions from a laboratory to plant size. That means we can just enlarge the laboratory equipment of the mixing to the pilot scale or industrial scale of production. But the same thing cannot be applied for the tablet compression, which involves the increasing production by increasing the speed. That means speed of the turret, speed of the filling of the die cavity. All these are going to be very critical parameters for increasing the production of a pilot from laboratory scale to the pilot scale to the industrial scale. So before going for any pilot scale up study, as I already mentioned, we need to follow certain regulatory guidelines. So European Medical Agency has given a guidance for process validation, and they have defined 3 different batch sizes, namely laboratory scale, pilot scale, and production scale. So laboratory scale is related to the research and early stage of the development of the product. Pilot scale is connected with the process development optimization stage, and production scale is connected with the real-time production batch. So the laboratory scale batch may be 100 to 100 to 1000 times less than the production scale, whereas the pilot scale, at least 10% of the production scale batch or minimum of 100,000 units, whichever is greater. And normally production scale batches are as per the contemplated production size of the respective industry. So the object of laboratory scale is to support the formulation packaging development, early clinical and preclinical stages. And it is a pilot scale also supports the preclinical and mid to later stage clinical evaluation and formal stability studies. So in the laboratory scale, we need not think about the stability, but in pilot scale, it is a must to go for stability studies. And it can be analyzed to assist the evaluation and defining the critical quality attributes, which we call it as CQAs in most common way. So in laboratory scale itself, what are the critical quality attributes that are to be addressed, must be taken care of. And in the pilot scale, we have to define what is the size, depending on the anticipated product volume, what are the constraints of the equipment, what are the regulatory expectations. So before going for a scale up, one has to question themselves, what is the objective of this pilot scale up? So is one model sufficient, or should tests be carried out in different models of different sizes? For example, a mixing operation, there are different types of mixtures. So we may be using the same model of the mixture right from the laboratory development to the industrial production. Or sometimes we need to change the model of the mixture or the type of the mixture that is going to be used. So one need to answer this particular type of model that is going to be used during the process. How small can the scale-up model be? This is very important and critical because the economics, more the size of the scale-up, more the cost that is going to be involved for its production. When must or when can physical properties differ? So depending upon the operation that is being subjected for the scale-up, the physical properties of the materials may change during these operations, and how we have to address must be taken care of. Which rules govern the adoption of the process parameters in the model measurements to those of the full-scale plant? So we have to know about the process parameters and the rules that are going to influence, because there are some mathematical equations using which we can efficiently calculate the outcome of a product that is being applied for the pilot scale or full scale, which I'm going to discuss shortly in the coming slides. Is it possible to achieve complete similarity between the processes in the model and those in its full-scale counterpart? If not, how one should proceed? So we have to have a similarity between the processes in the model that is developed for the development of a product and which is going to be taken for a full scale up. So if there is a difference, how we have to address these differences and how we can bring the similarity between these models must be addressed. And besides this, as I already mentioned, the regulations, scale up and post-approval changes, SOPAC, which is a well-known parameter for many of our regulatory students also, which we are going to teach. So this is to be addressed properly. Any significant change in a process of making a pharmaceutical dosage form is a regulatory concern, as I already mentioned. So FDI is coming with various documents on SUPAC, and they have already framed the guidelines for immediate release solid oral dosage forms, modified release solid oral dosage forms, semi-solid dosage forms. And they are going to develop the regulations for transdermal delivery systems, bulk actives, and sterile active solutions. The meaning of this is the SUPAC is going to be very, very important. That means for already existing products, whenever they are going to make any small change in the master formula record, that should be approved by the proper regulatory authority, and then only the product is allowed to come into the market. So, what is the origin of this? Product Quality Research Institute has been launched in 1999 with collaboration between the FDA, the pharmaceutical industry, and academia. And it is a non-profit organization working together to generate and share the relevant information for advances in drug product quality and development. So post-approval changes in the batch size is required to be submitted with additional application with specific requirement. We have to justify why we are changing these method modifications from the original record, and is it compliance with the CGMP and existing SOPs. So sometimes we have to give the data related to the new stability after the making changes, new dissolution information, and sometimes they may ask even the in vivo bioequivalence testing, especially whenever there are changes depending upon the levels that are done. That may be level 1, level 2, level 3, etc. And from level 2 onwards, if there is any equipment change, what is the reason for the different designs, etc. Level 2 process changes or level 3 process changes, one has to substantiate. And we have to do this documentation, which is going to be very costly. So way back from 1977 onwards, the FDA Office of Planning and Evaluation studied the impact of SUPAC guidance on industry, and the findings have given some substantial savings and permitted, because shorter waiting times for the site transfers, more rapid implementation of process and equipment changes, increases in the batch size, and reduction of quality control costs. So these are the advantages that are derived by this initiative of the FDA Office of Planning and Evaluation. So coming to the process of pilot scale-up of tablets. So whenever any tablets are being manufactured, the first and foremost problem that is encountered during the scale-up is mixing. Because we are going to mix the active pharmaceutical ingredient with various excipients to make the powder blend in the form of a wet granules or a dry granulation, whatever that is. So it is going to have a homogeneous blend. That is the whole object of this mixing operation. And we need to determine the composition of the mixing throughout the blend. And sometimes it is very difficult to have an online process, because we have to take the samples from different parts of the mixture, and that is to be analyzed for the homogeneity of the mixture that is being prepared. So there are some assumptions. We can have small number of samples, because more the number of samples, more the analysis that is to be done. So we assume that the smaller number of samples can sufficiently characterize variability throughout the blender. A standard sampling technique retrieves samples that are truly representative of the local concentration of at a given location. This is second assumption. The degree of mixedness at the end of the blending step is a good indicator of homogeneity. So we are going to assume these assumptions. Assuming these assumptions, these samples are being analyzed, but that may lead to a lot of problems, which I'm going to define in this particular slide. So how we are going to define the mixedness and what are the limitations? So there is no general mixing index that is available for all the mixtures that are being prepared in the industry. So one needs to determine the homogeneous mixing by way of its own standards. And what is the sample number and what is the sample size? And that may influence the variability. And small samples may exaggerate the variation, while large samples can blur concentration gradient. So we have to have sufficient number of samples that should be taken for representing the total blender volume. And in assumption 2, we may be using the thief probes for taking the samples from different parts of the mixtures, and while introducing these thief probes, which may not go to the right sampling point and maybe causing the problem for collection of the sample. So we have to have a proper thief probe sampling procedures, and filling the cavity of the thief probe is also going to be very, very important, and we have to take sufficient care. And in the case of granular mixtures, there is a chance of segregation or separation, and we have the unlike composition, which may be caused by flow, vibration, shear, etc. So once a good blend is achieved, the mixer still must be handled carefully to avoid any demixing that might occur in this particular granular mixtures. So whenever we are using the mixing operation, tumbling blenders are the most useful mixers. So it takes by 3 mechanisms. One is convection, the second one is dispersion, and third one is the shear. So in majority of these blenders, these 3 mechanisms are very active, and very little shear is imparted in majority of these blenders, except where an intensifier bar or chopper blade is used in the blenders. So we cannot create a single mechanism of mixing like a convection or a dispersion or a shear. So they have their impact on the mixing rate. So we have to have the symmetricity. So the material is going to cross from one side of the symmetry plane to the other side of the symmetry plane. So many a times, if there is an asymmetry, they have very, very good high mixing proficiency. So it can be induced through intelligent placement of baffles. So in the V blenders, sometimes they create the baffles for the creation of asymmetry, and the symmetric plane is getting disturbed, and we can have more efficient mixing of the powders. And loading of the V blender is also very important, either through the exit valve or equally into the each shell. And how we are going to fill the V blender, that means 1/4, half, 3/4, or almost near to the brim. So this also plays a very important role, and depending upon the load that is filled into the V blender will have its influence on the mixing index value. So there is an equation, sigma square is equal to A e to the power of minus Kn, where sigma is the mixture variance, n is the number of revolutions, A is an unspecified constant, and K is the rate constant. So the rate constants for convective mixings are very higher compared to the dispersive mixings. So unequal loading across the symmetry plane places emphasis on the dispersive mixing and is comparatively slow compared to top to bottom loading, which favors the convective mixing. So please take my lecture in the positive side, because sometimes I'm giving small equations here and done, but don't get confused that I'm giving too much complex equations. Just I wanted to emphasize how we can use the theories that are proposed for the mixing or the effective pilot plant scale up or the production scale up batches by using these approaches. So the equation also explains the effect of process parameters like number of revolutions and its effects on the rate of mixing. Low fill level of the blender can increase the mixing rate than the high fill level. So we can assume, depending upon each mixture and its characteristics, we can have this equation applied, and accordingly we can derive the most optimum conditions of the mixing. So there is one more number called as Froude number, which is a dimensionless factor given by V square multiplied by R by G, where V is the rotation rate, R is the vessel radius, and G is acceleration due to gravity. And by calculating FR as scaling parameter. So when they want to increase this blender size to 25 cubic feet, the linear increase is calculated to be 71%. And for the geometric similarity, the fill level must remain the same. That means whatever fill level that is used for the 5 cubic feet blender, they have used the same fill level for the 25 cubic feet. So for maintaining the same Froude number, the R is increased by 70%. That means the radius has been increased by 70%, 71%. The RPM, revolutions per minute, must be reduced by a factor of 1.71 into minus 1 by 2, which is equal to 0.76. And accordingly, the speed of RPM must be reduced to 11.5 because which is the slowest RPM that can be selected for the sensitive blender speed that is designed. So in the initial stages of the 5 cubic feet blender, the blend time is optimized at 15 minutes at 15 RPM. Then the total revolutions of 225 must be maintained to scale up. So at 11.5 RPM to attain 225 revolution blend time, the blending time should be 19.5 minutes. That means in the scale up blender, the blending time is increased from 15 minutes to 19.5 minutes. So this is the applicability of this Froude number, and using which we can have, and as I already mentioned in my previous slides, this is all related to linear increase. So this approach is convenient but empirical. That means based on the mathematical calculations. So there are some common violations of this approach. So when we are going to do the scale up from one geometry to another geometry, like V blender to in-beam blender, changing the fill level without consent to its effect, keeping the blending time constant. In the previous case, we are not keeping the blending time constant, but in this case, we wanted to keep the blending time constant while changing the blender speed. So it seems it is a non-systematic means of scale up and often may lead to excessively longer processing times and inefficient use of the existing capacity. And it can also lead to unwanted side effects such as particle centering, heat buildup, attrition, and excessive agglomeration. So what is the new approach for the scale up? Just a small 1 minute. You sign on the documents. You sign the thesis. You sign it. You got the synopsis also? Yeah, you got. You sign this thesis. I'll sign it. Then you can go for submission. So we have to determine the particle size, particle velocities, flowing layer depth, and the amount of particles in motion. So whenever we are going to handle the granular system, we need to know all this information. And a systematic generalized approach for the scale up of granular mixing devices is still far from attainable. That means they are not fully established. So how to come out of these problems? So there are some researchers who have published some good number of papers, and I'm trying to consolidate all those things in the form of these points. Scaling should not change the mixing mechanism. That means whenever we are doing the scale-up process, it should not change the method of mixing either by using convective or dispersive or something like that. That means the convective should not be completely transformed to dispersive or dispersive to the convective. So when we are going to do all this, it can be done by using asymmetric mixtures. So number of revolutions is a key parameter, but not the rotation rates. For example, in the previous example, the total number of revolutions are kept constant at 225, but not the RPM. So in the smaller mixture, the RPM is higher. In the larger mixture, the RPM is lower. That means we are not going to increase the rotation speed of the mixture, but the total number of revolutions are going to be kept constant. The number of samples, as the scale up is getting increased, the number of samples are to be proportionately increased to have accurate description of the mixture state. How we are going to interpret the samples is also going to be very, very important. Mixing index, what we have to take into account is the way in which it is going to be analyzed must be uniform throughout. What is the fill level and how it is to be filled. That means decreased fill levels or increased fill levels may cause dead zones and it may cause improper mixing. So the ultimate solution is addition of asymmetry into the vessel, either by the design of addition of baffles, will have a tremendous impact on the rate of mixing of a powder. The second important problem that is faced during the scale-up is handling of the powders, because segregation is a very big threat for the handling of the powders and it may result in a poor and incomplete blending. So we have to have a reliable flow for the powders, and when they are having a reliable flow, we have to maintain a blend uniformity. We can write mean right? A small interruption may be there for 5-10 minutes. Please bear with us. So the problems that are encountered in the flow of the powders are the arc, rat hole, erratic flow, flooding, or uncontrolled flow. So there are two types of powder flows what are going to be observed when they are being handled through bins or hoppers. One is called as funnel flow, the other one is called as the mass flow. So whenever a powder is flowing through a funnel, a channel is going to be created above the outlet and which is surrounded by a stagnant material during its flow. So when the powder level decreases in the funnel, the stagnant powder may also join along with this flow channel, and if there is any cohesiveness to the powder, it may create a rat hole like pattern. In the case of mass flow, all the powder will be in the motion. So in the case of funnel flow, part of the powder will be in the motion and part of the powder in the stagnant way, which is adhering to the funnel walls. In the case of mass flow, there is no such adhesion and all the powder will be in the motion flow. And in this case of mass flow, both in the case of funnel as well as mass flow, first in and first out flow sequence is going to be observed. But in the case of mass flow, it is much more predominant and it eliminates the stagnant powder. And it provides a steady discharge with a consistent bulk density and provides a flow that is uniform and well controlled. So in majority of the flow patterns of the powders, people will always prefer to have mass flow rather than funnel flow. So the importance of mass flow can be very much applied by designing proper outlet size of the funnel, which prevents the formation of the arch. How the powder flow properties are going to be influenced. So the flow properties of the powder may be influenced by the cohesive strength, wall friction, and bulk density of the powder. So cohesive strength may be resulting due to the arching and rat holing, and we can directly measure by using a shear method. And we have one more parameter called as flow function, which is going to describe the relationship between the cohesive strength of the powder and consolidating pressure. And in the case of wall friction, friction of the powder and the wall surface in contact with it is going to be the parameter that is going to be measured. And we can just measure it by sliding a sample across the stationary wall surface of the particular surface through which the powder is going to flow. And it may be also used by shear tester, and it is also used to determine the hopper angles. So what is the angle of the hopper through which the powder is going to be flowing is also going to be very important for assessing this wall friction. So bulk density, it also varies with the friction of the consolidating powder. We all know what is the use of the bulk density, hence I don't want to take much time on this. Why we are going to talk more about the powder flow across different bins and hoppers? Because the major problem I have already mentioned is the segregation of the powder. So segregation is the unwanted separation of the different components of the blend, and it can take place whenever forces are applied to the powder by way of gravity, vibration, or airflow. and they are going to act differently on particles with different physical characteristics. And the characteristics of the powder may include particle size, shape, and density. So how the segregation is going to happen by these three operational parameters like air entrapment, particle entrapment, sifting, and percolation. So to avoid the sifting and percolation, the particle sizes must be in the ratio of 2:1 is more required, and mean particle size should be not more than 100 microns. And the mixtures must be relatively free flowing, and there must be relative motion between the particles. And smaller, in the case of bin handling, the smaller particles will be concentrated under the fill point, with the coarse particles concentrated at the outside of the pile. So whenever the children are playing near the beach, you can see the larger, coarser particles are going always to the bottom side, and finer particles are going to separate on the top of that. And in the case of air entrapment, whenever fine and aerated powders are being handled, with the vibrations that are caused in the that are there, with the variations that are there, the particle size or the particle density, the vertical striation pattern is going to observe, and the finer larger particles concentrated above larger denser ones. And in this fluidized state, larger and denser particles tend to settle to the bottom. And particle entrapment occurs primarily with fine powders that are varying in their particle size or density, and they remain suspended in the air for a longer time, and their larger particles will concentrate near the incoming stream, whereas the smaller particles will be transported into the slower moving air. So how we have to identify this segregation problem? So whenever we are going to handle the powders at the bench scale, we can use two qualitative indicators. One is to sieve the powders by using the different screens and by measuring their particle size. But here the major problem is, whenever we are going to use the screens, the rotation of the screens may cause more vigorous separation than what is expected. And the second approach is the segregation test. And in this, we are going to subject the powder for the real application of these forces that are responsible, and how we are going to apply these forces for the segregation is going to be very critical in its evaluation. And in the pilot scale or the production scale, they are recognized by comparing the standard deviation of the particle size of the final product for those collected either within a blender or upon blender discharge. That means we are going to collect the samples from the blender directly during its operation, or whenever it is getting discharged from the blender, the samples are collected from different ports, and they will be analyzed for the uniformity of the particle size, and accordingly they are going to assess whether there is any segregation or not. Sometimes we will be using the stratified sampling technology, and that is going to correlate it with the flow pattern of that particular powder. So the approaches that are used for avoiding segregation are: modify the powder in a way to reduce its inherent tendency to segregate. That means we have to change the particle size distribution, change the particle size such that the active segregation mechanisms becomes less dominant, change the cohesiveness of the powder, increasing the tendency of one component to adhere to another. And the second approach is modification of the equipment to reduce the forces that are responsible for segregation. So minimizing the number of transfer steps, minimizing the drop height, that means from which height of the funnel the material is being dropped into the next operation, control dust generation, control fluidization of the powder. Slowing the fill rate can reduce fluidization and dusting segregation, and creation of a separate pathway for vent line for air to escape without moving through the powder bed, a deflector or distributor for the material stream as it enters the bin. Press hoppers, transfer chutes, and Y branches must be designed correctly to avoid the stagnant material. They should not have any crevices or obstructions for preventing the powder flow. Butterfly valve should be operated in full open position, not throttled to restrict the flow. And reblending of the powder during subsequent transfer is another approach. So whenever a powder is suspected to have been segregated, we have to have a reblending during the discharge. That means at the final stage of the discharge, again the powder is subjected for reblending. So there are some material instruments that are designed like bin set to do all these operations. And controlling the velocity profiles during the transfer is also very effective for preventing this segregation. And the third part is pilot scale of tablets by using high-shear wet granulation. So in this, the high-shear mixture force influences the particle consolidation and particle attraction for the formation of these granules. So whenever the pilot scale-up of tablets are going to be taken, normally high-shear wet granulation is going to be the most conveniently used process in the industry. And that is going to be decided by the geometry of the mixture, speed of the impellers, angle of the impeller blade. What is the acceleration force, resistance of the bowl, centripetal force, centrifugal force, gravitation force, fluidization force. And they determine the reproducibility of the resultant granulation process. So these are all the parameters that are to be taken care of whenever any high shear wet granulation process is being applied. So we have to have a design of equipment that maintains the geometric and kinetic similarity, and it is not that much easy to design the equipment for maintaining the similarity from the laboratory scale to the industrial scale. So we cannot duplicate the process in majority of the cases, and it should be tailor-made. So we have to use the dimensional analysis for coming out of this particular problem. And we have to define what is the tip speed of the mixer plate, what is the Froude number, and what is relative swept volume RSV. So tip speed is defined as flow RPMS into 2R, that is radius. FR already have given it, and RSV is the volume of material swept per second by volume of the bowl. So these are all the three different parameters that are going to be evaluated in the high shear mixtures. I'm just giving you some examples between the different mixtures that are used in the industry. So in the case of collet mixture and fielder mixtures, based on the bowl capacity and the tip speed, the fielder mixture is giving more mixing ability compared to the collet mixture. And they calculate the Froude number at low flow settings compared to between these two mixtures, and the graphs are presented. Let's not waste the time on this. So these are all the outcomes of this particular study. As the surge of the bowl increases, the FR decreases. So FR represents the compaction force during the granulation, and smaller development equipment with high FR have a tendency to form larger and denser granules, and also sticking to wall is greater. FR can be kept constant during scaling by increasing the speed of the flow with increasing the bowl size. So they also compared one more mixture along with this between the relative swept volume and bowl capacity, and the diazona mixture has shown more efficiency compared to the remaining two. So this is an example what I'm presenting here for this. In this, they have prepared the batches of 25 liters, 75 liters, 150 liters, and 300 liters high shear mixtures with the same formulation, the same type equipment. So when they are comparing the different equipment, they have taken the size distribution for the sustained release matrix formulation. And if you can look at this, the size log geometric log normal or geometric mean remained almost constant, and almost the other parameters like bulk density, tap density are also very near to each other. And that indicates that by keeping the F number constant, we can have the same parameters for the process batches which are taken from the laboratory to the pilot to the industrial scale. Just for 5 minutes, I'll take a small break. You got the CD?
Yeah, I got the CD. That is the break. That is not formed properly.
Huh?
That is the deform.
Okay. 20?
27.
Take the print of this there on there. I think I have kept the original. I have written keep the original books. Right? Give me the other pieces. You got the original? Check the date once again. I have to get back here. Where is the CD? You can write the CD. Once it is burned, huh? Just check that seal. Open it. Right? Yeah, sorry for the break. I have to just sign some papers of my student. Yeah. So whenever the earlier discussion we have seen, we are using the similar type of the blender or for making the scalar. But sometimes it is going to happen in such a way such that we may be using the dissimilar equipment. So in the case of scale-up that is going to be adopted by using the dissimilar equipment, we have to use two different approaches. The first one is called as the trial and error adjustments. The second one is called as the empirical correlations. So in this trial and error method, we'll be using the quantities that are to be asked as used like a granulation liquid amount, rate of addition, kneading time, etc., etc. unless we achieve the satisfactory product. And we can evaluate these granulations by evaluating the particle size distribution, bulk and tap density, and visual judgment if they are experienced in doing that. And in the empirical correlations, which is going to be a curvilinear plot or by advanced statistical equipment, what is the power consumption that is going to be used for the particular operation between the dissimilar equipment? What is the type of the parameters that are going to be associated for reaching to the endpoint? All are going to be evaluated, and they are going to be scaled in a linear way to arrive the required additions of the parameters. So majority of the time, they'll be using the process analytical technologies for control of these parameters in the empirical approach, where they are going to establish a parameter by using these process analytical tools, and they correlate the properties of the raw materials, and they are trying to understand the granulation processes. In some cases, in the PAT technologies, they'll be using the visual imaging technologies or light scattering technologies for monitoring the growth of the granules to terminate the process after reaching the predetermined particle size. And during this process, they collect a lot of information which can be applied to the other batches. But in this case, the major advantage is being the time. In the previous trial and error batches, more number of trials are going to be taken up for the scale-up production. Whereas in this case, based on the earlier data that is available, they can reduce the time significantly. So this is an example for the PAT technology application where they try to measure the particle size growth using the real time by using a visual high magnifying camera, during which time they started collecting the samples at different size. They started measuring the size of the different particles at 1632, 1633, 1635, 1636, like that. And they are going to measure the average particle diameter at different speeds and different values. And they also measure the torque of that particular equipment. And they try to plot a relationship between the particle size and the real time that is applied. So a case study in which a wet granulation process was scaled up and a high dose drug, almost 75% drug loading, was prepared for the wet granulation, and they tried to optimize the critical formulation process and process. Sorry. Yeah. They tried to optimize the critical formulation parameters and process variables. And before the scale-up batches, they tried to prepare the pilot batch with a 40 kg scale, and they tried to show the robustness, processing variables, etc. And based on that, they tried to optimize the parameters. So this is the table in which I tried to present this information. So the 40 kg scale-up batch, they have done two cases. One is the minus batch, the other one is plus batch. That means after optimizing the conditions of the 40 kg scale-up batch, they have reduced the levels of the parameters like granulation time, water level, drying time, temperature, lubricant level, compaction force, compaction speed, etc. And they tried to establish a relationship whether this batch can withstand the lower and higher conditions. Yeah.
Only one CD.
Yeah. Yeah, you take it. This deform segregate. So after making this pilot scale-up batch, they optimized the formulation conditions for the different variables, and finally they arrived at the optimized formulation. And this is they tried to compare the hardness of these pilot batches at the minus side as well as plus side, and when the variables are changing, there is a slight increase in the hardness of the particular tablet batch from the minus side to the plus side, but that did not significantly influence, and hence they could optimize these conditions. So the advantages of this particular approach is by using this design of experiment, we can develop a robust formulation and a process can be stable, and it will be used for large scale production, and we can have acceptable hardness and friability during the large scale productions. And it also offers the production flexibility, and we can do some adjustments for the force of the compression, speed of the tablet machine, and we can have the batch to batch variability also can be accommodated. Coming to the scale up of the pilot scale up of the capsule dosage forms. Capsules are also very prominent, and nowadays they are regularly developed for the various applications. So we have two types of the automated tablet machine, capsule filling machines. One is called as the dosator type, the other one is dosing disc. So in the dosator type, the powder is compressed into flux inside dosing tube with spring-loaded piston before being ejected into the capsule body. In the dosing disc, it uses as a tamping pins to form the plug within the cavities of the dosing disc, and finally it is ejected into the capsule body. So depending upon the type of the machine that is going to be used, we have to have certain criteria that are to be followed for the flow of the powder. So a low lubricant level is required for the dosing disc machines, and higher level of compatibility is needed for dosator machine. So we have to evaluate carefully about these lubrication levels depending upon the machine that is going to be used. So as we already discussed about the problems of the powder mixing and segregation, one should take enough care such that there should not be any leakage of the powder from the dosator, and it may lead to subsequent weight uniformity issues. And sometimes the speed of the capsule filling machines are very high, and some of the machines are capable of producing about 200,000 capsules per hour. So during this particular high speed operation, there is every chance of segregation of the powders, which may lead to weight variation of the filled capsules. So we have to optimize this tamping pin compression force, tamping pin displacement, and plug ejection force, along with formulation conditions such as particle size, lubrication level, blend uniformity, which will be useful in development of a robust manufacturing process. Once a pilot scale-up is successfully done, for regulatory concerns, they should conform to the stability of the product during the storage. So we all know the ICH guidelines, and the primary batches should be of the same formulation and packaged in the same container closure system as proposed for the marketed formulation for carrying out the stability studies. So the manufacturing process must simulate the production batches and should provide product of the same quality, meeting the same specifications as that are intended for marketing. And normally we have to prepare 3 batches, and out of the 3, 2 should be at least pilot scale batches, and the third one can be smaller if proper justification is provided. And the third condition that is laid down is wherever there is a possibility of preparing this drug product, different batches of the drug substance, that means API, should be used. That means we cannot use the same batch of the API for all the 3 batches, but it is preferred to have 3 different batches for the 3 drug product batches. So we have to define what is the target product profile relative to quality, safety, and efficacy. We have to identify the critical quality attributes to study the control of the product characteristics, and we have to determine the quality attributes of the drug substance and excipients, and we have to select the appropriate manufacturing process for controlling the strategy. So for doing all this, the approaches that are followed are quality ensured by end product testing, full design experiments, and hybrid designs. So quality ensured by end product testing, that means our normal laboratory empirical approach. And here, the understanding of process is very minimum. That means we go with the established procedures. So, and we'll be conducting the research on a one factor at a time. And there is no design space at the end of the investigation. So sometimes we may not adhere to the manufacturing conditions, and there is a chance of variability. And critical process parameters are determined only based on the prior knowledge and proven acceptable ranges, which are not determined. So we have the minimum knowledge about product acceptable ranges on the product that is developed. So how do we do this end product analysis? We mix the drug and excipients. We use the different unit operations. We use the product analysis, and we define whether the specifications are reached or not. And if the specifications are reached, we say the process is closed. If the specifications are not reached, again, we restart the cycle until the specifications are reached. So like this, we prepare the final end product. But the major problem associated with the final end product is more number of trials that are going to be involved, and critical process attributes are not defined properly, and any small deviation in the conditions that are already developed may cause a lot of variation in the final end product, leading to quality concerns. So as per the ICH Q8 pharmaceutical development, there are certain guidelines which are to be followed, and if we follow this process, the product may be of good quality compared to the previous end product design, where there is every concern from the regulatory authorities that how you are able to reach your end product. We have to answer as per the new regulatory guidelines. So by using the quality by design approach, we are going to use the design of experiments for the formulation development for producing the quality data for the early stage and reducing the cost of production by reducing the number of steps. And the analysis of data of the QBD will be helping in defining what are the critical quality attributes of the drug product and what are the critical process parameters. And it can also provide insight into the final product, the processes, designs that are involved, and whether they are conforming to the proven acceptable ranges of the process parameters. So what are the targets of QBD? So quality target product profile is going to be defined related to therapeutic equivalence, pharmaceutical equivalence, and bioequivalence. And critical quality attributes are going to be defined related to quality, safety, efficacy, and multidisciplinary as per ICH. And quality risk assessment is being done by using the Ishikawa fishbone diagram and risk-based matrix analysis or failure mode effective analysis. And by using the design of experiments and design space for screening and optimization by contour and overlay plots. So why we need to use the QBD? So we have to design the process analytical technologies, and there are different software that are used for making this PAT. And by using these things, we can design what are the process analytical technologies that are to be implemented for the QBD. And we are going to design the control strategy. So we are going to control the critical manufacturing attributes and critical process parameters, etc., again by using the different software. And for achieving this, the approaches that are followed are full design experiments. So we are going to have an enhanced QBD approach, which includes a systematic evaluation. So we are going to identify the material attributes and process parameters that are going to affect the CQA. And we are going to determine the relationship between the material attributes and the process parameters to produce the CQA. And we're using the risk assessment tools. We are going to identify what is the risk potential of these parameters for narrowing down the number of experiments to be conducted for modeling a design space. So we are going to define what are the material attributes, what are the quality attributes, what are the process parameters for designing the required quality target product profile. And in the risk assessment, we are going to understand what is the prominent critical attribute that is going to cause the risk of this particular product to failure, cause the failure, or the parameters that are responsible for success of this particular product. So by using this Ishikawa fish diagram, we have identified various process parameters for production of a tablet, like what are the plant factors, what are the compression factors, what are the drying factors, what are the granulation factors, what are the raw material factors, and what are the analytical factors, and finally, how we can produce a quality product. And by identifying this, again, in the mixing, we have subdivided this Ishikawa fish diagram into the low risk based parameters and high risk parameters. And accordingly,
everything is still showing.
but you keep it in this and check it in this. So the step 3 is we have to assess the impact of these parameters on the production parameters, and we have to rank them in processing steps which are going to have the influence on the CQA. And we have to determine the parameters that are to be investigated experimentally, and we are going to list these parameters that are to be investigated in a systematic way, which are to be screened. And what is the design space? That means what are the input parameters and process parameters that are to be considered for the design of these particular experiments. And we may be using different levels of the designs, like a 3-level design. And by using the response surface method, we can calculate what is the design space. But the full designs are going to have full designs need more inputs of the information, and sometimes it may not be easy for the researchers or the scale-up scientists to take up all this information. So as a via media approach, a hybrid method is developed. So we have to prepare more number of batches in the full designs, whereas in the hybrid approach, we are going to combine the initial 2 approaches. That is the first trial and error approach and the full design approaches, which is going to help in optimization of both time and cost, and it may also give good insight into the manufacturing process. That means we have to balance between the development time and cost, and it can be achieved by breaking the manufacturing process into 2 defined sections. So we have to prepare an initial blending step. For example, an initial blending step can be performed using end product testing, and the blend could be manufactured to specification, and end product testing will show the quality. For example, because it is not that much a serious issue, by using the end product testing, we can prepare the initial blend that is suitable for further processing. So the next processing involves the dry granulation, followed by compression into tablets. And in this, we may be using a full design experiment where the parameters that are going to influence the tablet manufacture can be taken into account for the design of experiments, whereby we can save a lot of time and money also in some cases to meet the target timelines. So what I would like to conclude in my presentation is we have to have a rational approach to pilot scale up, which is based on process similarities between the different scales, and which can be achieved through dimensional analysis by using the equations which are mentioned in the earlier slides. We have to eliminate the potential scale-up problems for development of these formulations such that they are robust enough for withstanding the processing conditions. And quality by design adoption in any scale of production is going to be very useful for ensuring the robustness of the product that is going to be developed from pilot scale to the production scale. And it also helps in determining the process parameters with their proven acceptable ranges. And in this particular case, I just give you a small example which was faced by one big company in Visakhapatnam when they are doing the commissioning process. So initially, it is a parental company in which they tried to use the silicone oil for coating the inside of the ampules for preventing the adhesion of the emulsion that is being prepared by them. So in the initial developmental stages, they have added this particular silicone oil to the ampules, kept it for 30 minutes, and again they washed these ampules, and finally when they analyzed, they found a value of approximately 0.6 ppm in the ampules, and accordingly they defined their internal quality standard as 1.2 ppm for each ampule. And later on, when they have done these production batches for the stability, as I mentioned, pilot production batches, the value is the values I have given it wrong, the other values I am giving it. The initial values, they found it as 0.01 ppm, and accordingly they fixed the value as 0.06 ppm. But when the production batches are taken up and they are analyzed, 5 minutes, I'll conclude. 5 minutes. When the production batches are analyzed, the silicone oil content in the ampules are going to be very, very high. So almost 10 to 15 times higher. So they have broken their heads. And finally, when they have gone into the documentation and videographs that are taken during this operation, they found that the operator responsible for closing the silicone oil and opening the vial has not done it properly. And in the initial validation process, he has not added the silicone oil at all for the soaking, but initially that was not opened. But during the operation of the production batches, they have opened it, and accordingly, the values of the silicone oil in the ampules or vials got shot up like anything, and all the 3 stability testing batches were rejected by the US FDA, and again, they have to submit a correction document for getting the approvals. And finally, it took about 6 months to 8 months for them to reach back to their production approvals. So a small mistake that is being done by a small person in the operation of a particular batch may cause a lot of havoc to the company. And by using these types of approaches of quality by design and all these things, one can ensure the robustness of a particular product during the pilot scale-up or industrial scale-up, and that will help to the company to a great extent. So these are all the references what I have taken for preparation of this presentation, and I thank one and all by namaskaram to all of you, instead of saying thank you by shake hand, because of the COVID pandemic, which is again like a volcano. It is spreading to entire India. So I request all my friends who are listening to me to be very, very careful by wearing a mask and keep safe distance from other known persons also. Don't be negligent in interacting with people while they are not wearing the mask. So I thank the Chebrol Hanmaya Institute of Pharmaceutical Sciences, Principal Vidyadhar, and other faculty members, the management of Chebrol Hanmaya Institute of Pharmaceutical Sciences, for giving me this excellent opportunity to share my thoughts on this pilot scale-up topic related to the AICTE sponsored staff training program. And I thank you one and all for the patient listening of approximately 1 hour. Thank you very much.
Thank you, sir. Thank you for your detailed talk on pellet plant scale-up. Thank you so much, sir. Dear participants, the next session will be started at 12:10 p.m.